Yes, in this fourth lecture, we will be looking at the plasticity, gastrointestinal plasticity during migration and during diet switching in birds. Gastrointestinal plasticity in endotherm vertebrates may not be like the gastrointestinal plasticity we see in snakes. The GI tract in mammals and birds responds to the diet, responds to food intake, responds to seasonal changes, but it does not do that the same way than the reptilian gastrointestinal tract. As I said earlier in the previous lecture, the plasticity of the epithelial mucosa is due to changes in cell number. Remember basically the mitosis in the deep, on the depth of the crypts of Linköping, mitosis of these enterocytes that migrate down up to the villi, where through a process of apoptosis will disappear, and then you have the constant renovation of the gastrointestinal mucosa. In reptiles instead you have a transitional type of epithelium that does not undergo this process of mitosis and apoptosis. So the process of renovation of the gastrointestinal epithelium in reptiles is different. The reptilian transitional epithelium is actually regarded as the ancestral trait that then in birds and mammals has taken a new form. In birds and mammals, this GI plasticity takes a different form in the form of cell proliferation and cell apoptosis. And what I want to show you is how actually birds, or how birds do when dealing with changes in diet and with migration. Some beautiful studies have been done in actually quail, and what you see here is basically how does the gizzard is the muscle stomach in birds, how does the gizzard change in mass as the animal changes the diet? Contains lower fiber with an increasing fiber in the diet. And what you see is very clear. The size or the mass of the gizzard increases more than doubles actually when you give more fiber. The understanding is that more fiber in the diet requires the muscle gizzard to basically mush the food much more. And as a result of this mashing, as a result of this effort, the gizzard increases in size up to 2.5 times. Notice that once you reach fiber concentrations of about 45%, there is very little change. And then what's interesting is that countering that, basically you have a decrease. You have a decrease in the liver mass. So the gizzard increases, the liver decreases. And this actually happens within 5 days of the diet switch. Even more interesting is the next. In this case, you have essentially the same animals, quail, that are actually fed for 2 weeks a high fiber diet, the 2 subsequent weeks a standard diet. High frequency, high fiber, standard diet. High fiber, standard diet. What is that you see? That the gizzard mass basically follows that beautifully. From 3 grams, basically almost to 6, doubling, down, up and down, up and down. So the gizzard in this case is following, is tracking exactly the diet composition, the food composition. The higher the fiber, the more muscular, the larger the gizzard gets. When the food becomes, again, the standard diet that contains less fiber, basically a drop. So there is an important plasticity in the system associated with the food the animals are eating. This is the same data, just shown in a more fancy coloring, but it's exactly the same concept, it's exactly the same data based on this data. What's important here is that these changes can be elicited repeatedly. Of course, the quail is not the only species. A lot of different bird species have been looked at. These species are on another bird that actually, when fed pellets, in these periods the animals are fed pellets, what happens to the gizzard mass? Drop. Pellets are much easy to mush. This animal actually will eat mollusks, and notice, eating some sort of clams here, going from mussels to smaller mussels, again, the gizzard actually responds very nicely instructs actually the diet composition. If you look now at the gizzard, what is it that you see? In the histology of it, this would be your control gizzard, and then this is the gizzard back to standard food, and in between you have 4 days with high fiber, 10 days with high fiber. What you see here, these sort of elongated bits here, is the nuclei of the muscle fiber. You have much more nuclei here than here, or even here. What does that mean? That means that basically the cells, the muscle fibers, become larger. That's what we call hypertrophy of the muscle fibers in the gizzard. So the gizzard is responding to the stress of actually breaking down higher fiber food by making cells larger, by enlarging the muscle fibers. How about in migrating birds? Migration in birds takes place with relatively small digestive organs. There's a reason for it. The cost of flight, you don't want to actually invest heavily in digestive organs. So what's seen is that birds, when they start going migration, they have relatively small digestive organs. GI atrophy, therefore, is being speculated to reduce wing loading and facilitate flight. An animal that's lighter can fly cheaper. Then the question becomes, how do they deal with refeeding stopovers? If the animal basically down-regulates the gut, atrophies the gut becomes smaller because it's not going to be equally relevant while migrating. What happens to those migrants? What happens to those migrants that do take stopovers? Does it mean that when you stop over, when the animal decides to stop to refeed, it has to stay there for a long time in order to rebuild the whole gastrointestinal tract? Some studies have been done actually with this bird, the black cap because it has sort of a cap that is black on the head. Bird names are always like that. Black cap, well, it must have something in the head. What's known in the black caps is actually that intake rate is dependent on previous feeding history. And these are experiments that have been done in these birds. Animals that have been fed ad libitum, that would be your basic control, have a food intake at this level. Animals that have been fed for the same period of time but less than ad libitum. You know that ad libitum feeding means that an animal is given as much food as it will take. In this case, it's a third of ad libitum feeding. It can take still the same amount of food. But what happens with animals that have been fasted? For 2 days they were given food, but then the last 2 days they were only given water. What's interesting is that in these animals' intake rate, the amount of food they can eat after this period of fasting is reduced. This suggests that when the animal is fasting, there is a reduction in the gastrointestinal mass, and this reduction actually has an effect on the subsequent refeeding. When this animal wants to refeed, there is not enough gastrointestinal absorption area in order to feed sufficiently. An animal that has been refed, this is the opposite group, an animal that has been given 2 days of water and then 2 days ad libitum, back up to the same intake rate. This data is showing us that birds can do this, down-regulate the gut and up-regulate it again. But they do it surprisingly fast. Sometimes we have this idea that, okay, it may take up a long time, a lot of resources to rebuild the gut. What the animals are showing us is that that's actually not so. In a relatively short period of time, in this case it would be 2 days, the animal can put back up everything back to normal. We would not be able to do that, but again, we have not lived in these conditions either. These changes are actually tracked by the intestinal mass. The animals that are fasted have the smallest intestinal mass, but notice that it requires 2 days before you are back to the refeeding. Refeeding group and the feeding group are almost identical here. So the message is, changes occur, plasticity is there, but compensation can actually come back very, very fast. In this case, it's actually known that birds, although they do have smaller intestinal masses, they are also more efficient. And this is a comparison basically between birds in the triangles down here with non-flying mammals higher up. Essentially, the nominal area of the gut for a given size of animal is smaller for birds. Essentially, the idea is they are more efficient. And the question becomes, how are they more efficient? The little we know is basically that birds use alternative absorption pathways. In mammals, the absorption pathway means that nutrients have to be coming from this is the apical membrane of the enterocyte. Nutrients are picked up here, transported using transporters inside the cell, out in the basolateral membrane and in the blood. What's known is that birds can use what's called paracellular pathways. Paracellular pathways are those that actually pass between cells. How do they do that? We know much less about it. There's just some studies that are showing this. These are studies, relatively recent studies, actually showing that the fractional absorption in birds is as high as it is in bats. So what this study shows is that bats, the only flying mammals, can do the same thing that birds, but other mammals cannot do that. Paracellular pathways are favored by birds and bats when it comes to nutrient uptake in the gut. And that seems to give birds a higher flexibility for nutrient absorption. That's it. Thank you very much again.